Hey, why are you eating so much fiber?
I am trying to support my gut microbiome. They are my friends and support my health.
Why? I thought we were your friends.
Oh, I think she's referring to her microbial community inside her intestines. We all have bacterial communities that inhabit our intestines. We greatly affect their population while they also have a great impact on our health. The relationship between our intestines microbial community and our health is a very complex one. Our gut microbiota helps protect against enteropathogens, helps us digest and extract nutrients from our diets, and contributes to our immune response. Let's take a look.
This microbial community is diverse and happy. The host consumption of indigestible and digestible fibers are supporting the gut bacteria. We are not limited to the digestive enzymes that our body produces. Along with the microbes we host comes their genomes and therefore digestive enzymes. This works to the host's benefit.
This microbiome is a lot less diverse as there is not as much of a food source for the bacteria to thrive on. When we eat refined grains, like white bread, the carbohydrates are readily absorbed and there is little to no fiber present. No fiber means less food for the bacteria. Competition occurs and only a few bacteria dominate the community, resulting in minimal diversity. Dysbiosis, which is the disruption of the normal balance between the gut microbiota and host, has been associated with obesity, malnutrition, inflammatory bowel diseases, neurological disorders, and cancer. For example, Clostridium difficile, a normal gut resident, can cause disease when antibiotics diminish a stable adult gut community. Similarly, other Clostridium species are associated with disturbed gut in infants. Infants are more susceptible to bacterial domination due to an underdeveloped intestinal bacteria community.
Whoa, there's a community living inside of us? Why are they so different?
It's because of a bunch of factors. Those include genetics, environment, age, and diet.
Wild. Tell us more.
What we eat has a huge impact on our gut microbial communities. Carbohydrate polymers, fibers, both digestible and indigestible, have the biggest impact on our microbes. There are less than 20 glycosidases, which are enzymes that cleave glycosidic bonds making up carbohydrate polymers, identified in the human genome. Our gastrointestinal microbiota contains 150 times more genes than our own genome. So, just because we may not be able to break down a fiber, it doesn't mean our microbes can't.
Also, the metabolites produced as a byproduct of microbial fermentation have been shown to benefit us. For example, the microbes digest complex polysaccharides, and the byproducts of these biochemical reactions are mainly short-chain fatty acids. These include acetate, propionate, and butyrate. A small amount of short-chain fatty acids have been shown to cross the blood-brain barrier. Also, short-chain fatty acids influence GI epithelial cell integrity, glucose homeostasis, lipid metabolism, appetite regulation, and immune function. Another beneficial component is that common gut residents, such as E. coli and Bacteroides fragilis, are involved in the synthesis of various B vitamins, vitamin K, folic acid, and biotin.
Our gut microbiota also has a great impact on our immune system. Our health depends on its strength and well-being. There's a symbiotic relationship between the gut microbiota and the immune system.
What's symbiotic?
They work to each other's benefit. Think of them as friends. The gut microbiota deploys 3 mechanisms to defend against potential pathogens. First, direct inhibition of pathogen growth using microbiota-derived substances, such as the metabolites we discussed earlier. Second, nutrient depletion. Lastly, microbiota-induced stimulation of innate and adaptive immune responses. The microbiota in your gut supports the immune system as it defends against invaders, while the immune system is more of the peacemaker between the microbes and the host.
What do you mean by that?
The microbes need a place to live and thrive without any interference, while the host needs to be able to do their own thing without any problems.
What does the immune system do for the host and the microbes?
Think of your immune system as the leader of the body. In order to establish authority and protect the harmonious relationship between the host and the microbiota, the immune system puts two lines of defense, the innate and the acquired. So let's say you have a pathogen and it tries coming into your system. Once it enters, the pathogen soon learns that it has to compete with all of the other thriving colonies that have been living in your intestines.
So this would make the gut microbiota our line of defense?
Yes, that's correct. They're the first line of defense.
Oh, I see. We allow the microbiota to live in our intestines, and in return, the immune system makes the microbiota work to protect us. Makes perfect sense.
Yes, and to help increase your microbiota, you can maintain a good diet and avoid anything that works against your microbiota, like antibiotics.
Yeah, the use of antibiotics can help cause dysbiosis, which occurs when the community is more susceptible to having a dominant bacteria overtake the population.
Now that I have an understanding of this, it makes me wonder if there is any research being done.
Actually, there is. Did you know that the Institute of Health, or NIH for short, started a project to study the human microbiome for 8 years until 2016. It's called the Human Microbiome Project. There are so many studies now investigating our microbiomes. Advances in technologies such as whole genome sequencing and 16S rRNA profiling have allowed for more in-depth research on these complex systems. We are now able to sequence whole genomes, and from that, we're able to map out and specify our entire communities.
Yeah, many studies are done using animal models or analyzing human fecal samples.
Oh yeah, I read that use of CRISPR technologies comes into play as well. A professor from the FBNS department, Dr. Rudolph Barango, along with June Go, has published an article investigating potential ways to improve our gut microbiome. In their 2019 publication, The Enrichment of Lactobacilli in CRISPR-Cas systems could potentially be reprogrammed for genome editing. This could make lactobacilli a microbe that plays a role in the colonization resistance against pathogens.
So that means what is being done now is the editing of some members of microbial communities to make them less susceptible of succumbing to pathogens. This has been done mostly with lactic acid bacteria. In the future, it may be possible to see more specific antibiotics with the use of gene editing technology like CRISPR. Conventional antibiotics are nonspecific and wipe out bacterial communities. This can leave us at a state of dysbiosis. If we can create targeted antibiotics, the intention is to preserve our present microbial communities and only target the pathogen.
Now that we know all of this, let's make sure we keep eating lots of fiber to keep our gut microbes happy.